Ya estamos, ¿no? Ok. Hola a todo el mundo. Primero voy a hacer unas pocas palabras en español. Me voy a quitar la mascarilla. And now we'll switch to English for the international audience. I am very, very happy to introduce Luisa Cochella to this second webinar of our cycle Severo Ochoa. I know Luisa for a few years now from our postdoctoral years in the Oliver Hopper lab at Columbia University. But I will give you a few milestones about Luisa's career. Well, first of all, she's Argentinian. She was born in Buenos Aires, and she did her PhD at Johns Hopkins University under the mentorship of Rachel Green, where she was studying the fundamental roles of tRNA in translation and protein synthesis. And after that, she moved to Oliver's lab in 2006, where she started to use C. elegans, the worm, as a model to study microRNAs in their role in neuronal identity specification. She published several papers, including a beautiful Cell paper. And I would like to say that I was always impressed by the courage that Luisa had in the lab, always implementing the more novel and more cutting-edge technologies in the lab. After that, in 2013, she started her own lab at the Institute of Molecular Pathology in Vienna, the IPN, where she holds a group leader position. She continued to use C. elegans as a model to study transcriptional and post-transcriptional mechanisms involved in the generation of cellular identity. They developed very clever and useful methods to study microRNA function with a single cell resolution. And they have also brought very important concepts, particularly, and I think she will explain us today with more detail how cell memory can impact on terminal fate identity. And I would also like to say that she's been awarded with many different fellowships, but importantly, she was awarded with an ERC starting grant, as well as recently an Embo Young Investigator Award. So I think I won't take more of your time, and with no further ado, I will let Luisa tell us about this latest research. Thank you.
Thank you very much. Thanks a lot, Esther, for that very kind introduction, and also for inviting me and giving me this wonderful opportunity to share our latest work with you. I would have much rather, of course, done it in person. I have never been to Madrid. It's something I would really, really like to do, hopefully soon. But for today, again, it's a pleasure to be here and tell you about our work on transcriptional regulation and cellular diversification. So, let's see, there we go. Like many of you probably in this audience, we're fascinated by the process during which a single cell gives rise to a complex multicellular organism. And the aspect of this process that we're most interested in and that takes up most of our time is how does the immense cellular diversity that makes up these different organisms form during development. And to address this question of how different cell types form, we choose to work with this animal, the nematode C. elegans, for a number of reasons that I would like to go into a little bit more detail. So for those of you who do not know C. elegans, this animal in the adult stage has precisely 959 somatic cells. So within this relatively small and one might say that relatively simple animal, there is a very large cell type diversity. This animal has more than 100 different neuron types, different muscle types, different epithelial types. So I think the worm strikes a perfect balance between simplicity and complexity that makes the study of cell diversification really, it gives us very unique ways to study that process in the worm. The other aspect that makes C. elegans a fantastic model to address these questions is the fact that thanks to heroic efforts from John Sulston, we know precisely the pattern of cell divisions that gives rise to every single cell in this animal. That's also, of course, because this pattern is invariant from one individual to the next. And what that means is if I decide to study this particular neuron in C. elegans, I know that in every single animal, this neuron develops through this very particular pattern of cell divisions. So that gives us the possibility to track, at least anatomically, the development of every single cell, every single somatic cell in the worm. So this makes C. elegans a really fantastic model to address questions of cell diversification. And what we are mostly interested in is in the gene regulatory mechanisms that underlie this cellular diversification that happens during development. Specifically, within my group, we're interested in two broad questions. We're interested in how transcriptional regulation over time is used to generate cell type diversity, but also we're interested in how post-transcriptional repression, which is mostly, or in our lab, the focus is mostly put on a class of post-transcriptional repressors called microRNAs, how these additional layer of regulation is superimposed on transcriptional regulation to shape cellular identities. But as the title of my talk suggested today, the focus will be on this question of how does transcriptional regulation over time impact on the terminal differentiation of cells. And the story that I will tell you will deal with how transient transcriptional inputs can have an impact on the terminal differentiation program that a cell executes, and how two cells that should execute the same terminal differentiation program actually differ due to their different transcriptional or developmental trajectories. So before diving into that, I would like to remind everyone how different cell types are actually defined by the combinatorial action of transcription factors. And this is something that has been most deeply studied in the context of generating neuronal diversity. So here's an example from C. elegans, from a lot of work that has been done in Oliver's lab, in Oliver Hobart's lab, including a lot of the beautiful work that Esther did as a postdoc in his lab, where what we see is, what has been observed is that many different neuron types in the animal are specified by the combinatorial expression of specific transcription factors. And these transcription factors can be reused in different combinations to generate additional cell type diversity. And so what that means is that the combinatorial action of transcription factors gives an animal or any organism the possibility to generate a larger cell type diversity than transcription factors are available. And these are two other beautiful textbook examples of such combinatorial action of transcription factors, for example, during Drosophila development up here, but also during the development of the vertebrate spinal cord, in which morphogen gradients define the regional expression of different transcription factors. And with a handful of these transcription factors, given their unique intersection or their unique overlap in different parts of this developing tissue, different types of neuronal or glial progenitors are specified. So if we think a little bit deeper about what does combinatorial action of transcription factors means, essentially what I'm talking about here is the fact that the expression of most developmental genes is not activated by a single transcription factor binding to its enhancer promoters, but rather by combinations of transcription factors, such as in this example, in which this purple gene will not be activated if only the red or the blue transcription factor are expressed, but only in those cells where both transcription factors are present and able to activate this gene. So what are the molecular mechanisms that we know confer transcription factors the ability to act combinatorially? So there are some, the most obvious ones are, for example, transcription factors that form a dimer, and when they dimerize, they now can recognize a new sequence on DNA that individual factors cannot. Or transcription factors that have binding sites relatively close together on DNA, and by mass action are able to displace nucleosomes and access the underlying DNA to recruit polymerase and activate transcription. So these are all mechanisms that require the simultaneous binding of 2 or more transcription factors. There are also instances in which sequential binding of transcription factors is required to achieve this cooperativity or this combinatorial action. And that is the case of the very well described pioneer transcription factors, in which these factors are defined by their ability to recognize their binding sites, even within a nucleosomal context or within some sort of closed chromatin context, and thereby displace nucleosomes and enable subsequent transcription factors to also bind and activate transcription. So all of these mechanisms require that 2 or more transcription factors are present in the same cell at the same time to be able to act on the same piece of DNA. And because of that, this requires essentially within a developing organism, the spatial intersection or the co-expression of these 2 or more transcription factors. What I would like to discuss today is how this combinatorial action of transcription factors can be achieved over time, where 2 transcription factors are expressed at different time points. They're non-overlapping, they're never present in the same cell at the same time, and yet they're both required to turn on a particular gene, whereas a cell that expresses only that terminal transcription factor is unable to do so. And this has essentially important consequences for the generation of 2 different cell types. So that's a concept that I will talk about. Now, let me go into the details of the context in which we develop this concept and the cells that we'll be talking about and the genes that we'll be talking about. So this whole story started by studying a pair of neurons, a pair of sensory neurons in the nervous system of C. elegans, in the head of the worm. They're a pair of neurons that are largely morphologically symmetric, and they are defined molecularly by the expression of a transcription factor called Ki1. Ki1 stands for chemosensory deficient because a mutant in this transcription factor abolishes the ability of the worm to chemotax to a lot of different cues in the environment. However, what's surprising about these 2 neurons is that they are functionally asymmetric. The left neuron senses mostly sodium and other ions, and the right neuron senses mostly chloride and also other ions. And this asymmetry in function is determined, of course, by asymmetric gene expression. Among the different asymmetrically expressed genes in these neurons are genes of the guanylate cyclase family. These are receptor guanylate cyclases. As you can see here, GCY7 is expressed on the left side, GCY5 on the right side. And these genes encode for proteins that localize to the tip of the nose of the animal, and they're thought to be the actual sensors of these different cues. So these neurons are molecularly asymmetric, and that molecular asymmetry is defined by the asymmetric expression of a microRNA. This microRNA was found in Oliver's lab, and it was called LAUC6 for laterally symmetric, because in a microRNA mutant, these 2 neurons now become symmetric. So LAUC6 is expressed exclusively in the left neuron. These are, by the way, I didn't mention, the name of these neurons is ASC. They are anterior sensory neurons, letter E. And ASCL will be for the left ASC neuron and ASCR for the right ASC neuron. So LAUC6 is expressed in ASCL exclusively. However, what was very interesting in trying to understand how the asymmetry of these neurons arises was the fact that for expression of LAUC6, the only gene that we knew was important was Ki1. So Ki1 is necessary for the transcription of LAUC6 in ASCL, but Ki1 is present in both ASC neurons. So it couldn't be the sole determinant of specificity. And after searching for many other transcription factors that could be acting with Ki1 or that could be repressing LAUC6 in the right neuron, what we realized is that the answer to the solution lied in the lineage. So the left and right ASC neurons, sorry about that, have a very interesting developmental history in that the left neuron derives from this blastomere at the 4-cell stage called ABA, while the right ASC neuron derives from a different blastomere already at the 4-cell stage called ABP. Now, these 2 blastomeres are initially symmetric, but they are made asymmetric due to notch signaling, and they give rise to different parts of the animal. However, these sub-lineages within ABA and ABP will at some point continue the same developmental path, and they will give rise to a number of neurons on the left and the right side of the head of the worm, including the left and the right ASC neurons. So what was very interesting, and that was a very important piece of information that we knew thanks to the lab, thanks to work from the lab of Jim Preece, is that one difference between the ABA descendants and the ABP descendants is that there's expression of two T-box transcription factors in the ABA great-granddaughters, but not in the ABP lineage. So these transcription factors of the T-box family are actually a very recent duplication. They are totally genetically redundant, and so I will usually talk about them together, but essentially you need either one for function, so loss of function requires removal of both of them. And they're expressed, according to the Preece lab, they're expressed transiently during the development of ABA. Now, the very curious thing is that we found that those TBX transcription factors were necessary for the expression of LAUC6. And they were not only necessary, if we ectopically expressed those T-box transcription factors in the ABP descendants, we would now turn on LAUC6 in the right ASC neuron as well. So it seemed that they were sufficient also to act in combination with Key1 to activate LAUC6 transcription. The other important piece of data that I want to share with you is that we also knew that the expression of LAUC6 was determined by that intersection between TBX3738 and Key1, because Key1 is only expressed in the ASC neurons. However, if we ectopically expressed Key1 in these neurons that arrive from the sister lineage, we would now also enable activation of LAUC6 exclusively in the left neuron that now ectopically expressed Key1. So what we found is that you needed both TBX3738 and Key1 to determine expression of LAUC6. Now, the very curious finding that we made also already a few years ago when I was in Oliver's lab is that it seemed that TBX3738 might be acting directly on the LAUC6 locus already at this early time point. And that's because we found in the locus a very well conserved TBX consensus binding site that we proposed could be the site of action of TBX3738. And so based on these and several other experiments, we proposed a model in which the early action of TBX3738 on the LAUC6 locus could prime the locus for the later activation by Key1. However, in a lineage in which TBX3738 are not expressed, Key1 would be unable to activate transcription of LAUC6, and therefore the asymmetry in LAUC6 expression would be established and the asymmetric neuronal function would be established. So this was a model that we proposed back then, and it actually took us a lot of effort to figure out how does this work. And when I say us, I mean basically a team led by really two fantastic people in the lab, a PhD student, Julien Jarret, who will defend his thesis soon, and a previous postdoc, Thomas Daniel. So Julien and Thomas set out to figure out how is the integration of these two transcription factors achieved at the LAUC6 locus. So the first thing that they did was to tag the TBX37 or 38 loci with GFP and use these for pull down to try to assess whether TBX3738 were really directly binding to the LAUC6 locus, because we had really no evidence that this was the case before this. So one important thing is that, as I mentioned, because these two transcription factors are redundant, each transcription factor was tagged in the mutant background of its counterpart, such that the only source of TBX transcription factor in these embryos is the GFP tag version. So Thomas performed chromatin immunoprecipitation and sequencing on these two different strains, and I'm showing you here the aggregate of several replicates that he did for both transcription factors. And what he found is that these two transcription factors are able to bind to the Lausy-6 locus slightly downstream of Lausy-6, where we had predicted those binding sites would be. And if we do global analysis of where TBX is binding all over the genome, we find that it binds to a motif. We can derive a motif de novo from these binding sites that looks practically identical to the known TBX binding motif. So we think this is a good binding site, but we wanted to test the functionality of these. So in order to do that, Julien designed a strain in which he engineered a YFP in place of the Lausy-6 locus in the genome using CRISPR. And he saw that these, as many of the other transgenes that we have looked at, also is expressed exclusively in the left AAC neuron. But then on top of that, he also deleted the sequence underlying that peak that we observed by, or that we found by ChIP-seq. And when he deletes these TBX binding site, he completely loses expression of Lausy-6. So we knew that the TBXs were important for Lausy-6 expression, but now we know that they mediate their function through this one binding site that's right downstream of Lausy-6. Julien also assessed the functionality of this binding site by looking not only at the expression of YFP from the Lausy-6 locus, but by looking at the downstream activity of Lausy-6, which is essentially the development of the neuronal asymmetry. So in wild-type animals, this reporter is asymmetrically expressed. However, if you delete these TBX binding site, now the neurons become symmetric. So this is the key binding site through which TBX 3738 mediate the expression of Lausy-6. So once we had these GFP tagged alleles, this enabled us to go back and ask what I think is the critical question or was one of the critical questions going into this project, which is the expression of TBX 3738 really transient? So is it really separated from the action of key 1? It had been proposed by the Priest lab based on antibody staining that TBX 37 and 38 were expressed during this time point and during this time point as well. But after that, they didn't see any signal anymore. So the first thing we did was we also looked at our GFP tagged proteins. And when we did that, we saw a very similar pattern as what they observed. We see that there's very high expression in these two cell divisions. We see a little bit extra expression, one cell division after, and then after that, we don't see any expression anymore. However, of course, not seeing something doesn't mean that it's not there below the limit of whatever detection method we're trying to use. So we were not fully satisfied with this, and we designed a functional experiment that would allow us to say whether the activity of TBX 3738 is required beyond this time point during which we can clearly see its expression. So the experiment actually consists in the timed forced degradation of TBX 3738, which we can also achieve thanks to this GFP tag that we put on this transcription factor. So essentially, the degradation of GFP tagged proteins can be achieved using a GFP nanobody that will recognize the GFP and bring along a ubiquitin ligase adapter to promote the degradation of that GFP tagged protein. So the experiment was the following. If we degrade TBX at this time point, we would expect that indeed Lausy-6 is not able to be expressed at this later time point. However, if the TBXs are really not required at these later time points, then we should be able to degrade them at later time points without any effect on expression of Lausy-6 later on. So here's the experiment. So if the expression, and one important thing, the control of this degradation system, of the expression of the degradation system, is done by using a heat shock promoter. So we can time the degradation of TBX GFP. So if we do an early heat shock where we target the peak of TBX expression for degradation, what we first observe is that this embryo, for those of you who are not used to looking at C. elegans, it really doesn't look good. A nice C. elegans embryo at the endpoint of development should look like a nice little folded larva inside of the eggshell. This embryo looks clearly malformed, but it looks exactly like what we would expect from a TBX3738 double mutant, suggesting that our degron system is able to degrade TBX37, and I forgot to say this is in the background of a TBX38 mutant. We're able to eliminate TBX3738 function with the same efficiency as complete deletions of these genes. There's an additional control that we performed in this case, which is that we express the ubiquitin ligase adapter in the absence of the nanobody. And that, of course, these animals will undergo the same heat shock. They will express the same adapter, but TBX3738 should not be degraded. And these animals all develop morphologically normal, but they also all express LAUC6. So we have a degron system that works very well. So now what is the result of the experiment? If we degrade TBX 40 minutes later, 40, and that is roughly the duration of one cell division, what we see is that has absolutely no effect either on the morphology of the embryos or on the expression of LAUC6. So this means that even if there was some TBX left in the lineage that we cannot detect by fluorescence or antibody staining, these TBX transcription factors are not functional and they're not required beyond that time point of expression. So this was very important to us to tell us that the activity of TBX3738 is really temporally separated from Q1. One thing I didn't mention, it's exactly separated by 4 cell divisions. So TBX3738 are not necessary at later time points, but do they really need to act at that early, at the early time point when they are expressed? And to address this, we performed the following experiment. We asked whether if we were to provide TBX37 at different time points, at later time points, would it be able to prime and therefore activate the LAUC6 locus? So we did this in the following way. We had a strain that had the YFP engineered locus that Julian made, and that strain expresses TBX3738 endogenously. So that strain will normally activate LAUC6 expression in the left ASC neuron. But now on top of that, we add TBX under a heat shock promoter, and we will heat shock animals at different time points of development, and we will ask whether it's also able to activate GFP, LAUC6 GFP expression also in the right ASC neuron. If TBX is able, because this heat shock promoter is ubiquitously expressed. So if we heat shock embryos at this early time point or during this time window during which TBX3738 are normally expressed, what Julian observed is that these animals express LAUC6 in both neurons, suggesting that this early ectopic TBX3738 is able to prime LAUC6 for later activation by Q1. But surprisingly, beyond a certain time point in development, and in particular, if we focus now at this early time point at 300 minutes, providing TBX at this late time point is unable to prime LAUC6 or to give Q1 the ability to turn on LAUC6. So what this means is that the LAUC6 locus must be primed or activated by these early transcription factors if we want them to be expressed later on in development. And if we miss that time window, then the locus will remain inactive, even though transcriptional activators are provided at a later time point. So we wanted to understand what is it that TBX3738 do to the LAUC6 locus at this early time point that makes it competent for the later activation of that locus by Q1. And in order to do that, we set out a system with which we could separate cells from the different lineages, the different parts of the embryo, and look a little bit deeper into the LAUC6 locus to see what the chromatin status or what the DNA accessibility status of the locus in these different lineages would be. And so for that, Thomas generated a strain in which he labeled the ABA lineage, and he could distinguish that from the ABP lineage and the rest of the animal. And then I Ariane and Alex performed a TAC-seq, which was analyzed by Jinqui in the lab. And this is what we observed. So they separated first cells from the ABA and the ABP lineage at this 90-minute time point. This is the time point of development at which we observe the peak of TBX expression. And what I've done here is I'm showing you both the track for the ChIP-seq data I showed you earlier, where TBX bind, and I'm showing you here the TAC-seq data for ABA and ABP. And what I hope you can see is that there's a very clear, open, accessible region that completely overlaps with TBX, with the TBX binding site in the cells that derive from the ABA lineage, but not in the cells that derive from ABP. And here, just a little note, I left this other locus downstream, which has equal accessibility in both lineages to show you that the experiment in ABP actually worked very well. We don't see signal here because there's really no signal. These cells are actually extremely similar in their DNA accessibility profiles. They differ only in about 10% of accessible regions, and most of those regions overlap with TBX37 binding sites. So fine, this is what we might expect. If there's a transcription factor binding to this locus, we might expect that this locus is accessible. Now, the very intriguing thing was that when the guys sorted cells from the 200-minute time point, at which we know TBXs are no longer there, and yet we still observed that the Lausisix locus was differentially accessible in cells that derived from either ABA or ABP, suggesting that this initial differentially accessible state that is determined by the presence of TBX is somehow still there, even though TBX 37 and 38 are gone. And now let me finally show you a data set that was obtained from sorted left and right ASC neurons. So this is sorting the mature neurons. And what I hope is very clear is that, again, there is a clear differential accessibility of the Lausisix locus in the left versus the right neuron. However, this region of accessibility is now shifted such that it's moved from the downstream binding site to an upstream region. And what I want to highlight is that this region of accessibility actually overlaps with the key1 binding site. So what we think this reflects is that in the left ASC neuron, where the locus has been primed, key1 is now able to bind and promote transcription, whereas in the right ASC neuron, even though it expresses key1 just fine, key1 is unable to do anything on this locus. So this was very interesting. So TBX 3738 seemed to be, and maybe one short experiment, one additional control that we did that might be useful to know about is that we also did a TAC-seq in a strain in which we deleted the TBX binding site. So this region of accessibility, as you might expect, is completely gone, but also there's absolutely no increase in accessibility around the key1 binding site and around the Lausisix promoter in the ASC neurons in the absence of that initial binding. So what this told us is that TBX 3738 are establishing an accessible state on the Lausisix locus, and we wanted to understand how do they do that. So they are transcription factors, so we asked, do they promote transcription? This is something that I want to spend a second clarifying, because we only see when we use reporters for Lausisix expression, or when we measure the production of Lausisix, which is a microRNA using different means, we only see expression really in the mature ASC neuron. However, what we're going to ask now is whether transcription might start already at these early time points, although at low levels, in depending on the presence or absence of TBX 3738. So in order to ask that, we need a more sensitive method, because clearly we don't detect very robust expression of Lausisix at those early time points. So Julien performed single molecule in situ hybridization, single molecule FISH, and that is achieved by having multiple short probes, each one with a fluorophore, that in our case were directed to a sense transcript, but also to anti-sense transcript. And just here as a technical note, because the microRNA locus is relatively short, we had to do this on a YFP engineered locus that produces a longer RNA transcript. So what you see when we look at the sense single molecule fish is that, so this is an embryo at a late stage in which there's very clear signal in one cell. This is the left ASC neuron, which is full of transcript once it's born. But if we go back and look at earlier embryos, we can see that at very early time points, this is shortly after the onset of TBX expression, we can already see a few molecules of transcript. And we can see transcription really throughout development in a manner that I'm not showing you here, in a manner that's fully dependent on TBX 3738 and also on their binding site. If we delete those, we completely lose all of this transcription. Now, interestingly, there's also, and potentially even at higher levels, transcription in the antisense direction. So this is what we're looking at here. What we see is characteristic bright foci in the nucleus that we think mark the sites of transcription. And we see those really throughout the development of the animal. And this is also, again, fully dependent on TBX 3738 and on their binding site. As the animal develops and in the mature ASC, this transcription is eventually gone. So there is transcription of the locus at low levels, but already at these very early time points. Now, the question was, is this simply a byproduct of two transcription factors binding to this site and doing what they're supposed to do, which is to activate transcription? Or is this transcription functionally meaningful? Do you need the locus to be transcribed in order to set up this primed state that will be competent for transcription, for robust transcription by key 1 later on? So in order to address that, Thomas generated some strains in which he deleted the TBX binding sites, and he replaced them by the binding sites for the yeast transcription factor GAL4. These are so-called UAS sites. So such a Lausisix locus in which there's no TBX binding, as based on everything I've told you, should not be expressed. And that's in fact the case. That locus does not produce any GFP at all. However, we asked what would happen if we now bring back transcription. And we did that using, again, the GAL4 DNA binding domain, which will be recruited to these binding sites, and bringing a transcriptional activator, in this case, VP64. And what we saw is that if we express this fusion protein, this GAL4 VP64, under the TBX37 promoter, so we provide this activation early on and in the ABA lineage, we now fully recover expression of this reporter in the left ASC neuron. However, if we bring back only the GAL4 DNA binding domain that should bind to this locus but is unable to activate transcription, then there's absolutely no GFP expression. So this was already a very strong indication that transcription might actually be necessary. So the other experiment that Julian and Thomas did to look at this from another angle was to take a strain in which the TBX binding sites are there, but introduce the UAS, the GAL4 binding sites, right next to the TBX binding site. So this reporter is now normally expressed, and that is true. If we tether to these binding sites just the GAL4 DNA binding domain, nothing happens. That locus is still perfectly expressed. But if we now tether next to the TBX binding sites a transcriptional repressor, in this case it was the C. elegans homolog of Groucho, UNC37, then we almost completely abolish expression of GFP from this locus. So what this told us is that the establishment of an active transcriptional state is actually really important. This is probably what defines the primed state of Lausisix. So one important experiment that we did to validate this heterologous activation system by GAL4 VP64 was to ask whether it behaved the same as endogenous TBX 3738 in their ability to activate the Lausisix locus over time. As I told you earlier, the endogenous TBX 3738 need to act during an early window of development in order to prime Lausisix for later activation by Q1. And if you miss that window of opportunity, even if you provide these transcriptional activators, the Lausy-6 locus is not expressed. So we did the same type of experiment I showed you earlier, now with the GAL4-VP64 system and the Lausy-6 locus that contains the UAS binding sites. And here are the results of those experiments. Again, if we heat shock, if we provide the GAL4-VP64 activator early on in the embryo, we see that in every embryo we looked at, we can activate expression of Lausy-6 also in the right ASC neuron. However, beyond a time window that looks extremely similar to the one we saw for TVX3738, providing GAL4-VP64 is unable to drive expression of Lausy-6. So what this told us is that the priming of Lausy-6 needs to occur at an early time point. And this is not, very likely not due to the nature of the transcription factors doing the priming, but rather due to some inherent property of the locus itself. And this is something that we will explore further in the future. So everything I told you so far led us to think that TVX3738 are acting early in development to prime the Lausy-6 locus, and this primed state can be defined as a state with active transcription, accessible chromatin, and that this is important somehow for the later boosting or activation of the locus by Q1. So what I would like to do with the next 3 pieces of data that I will show you is try to address the question of how is that primed active state that is established early maintained during 4 cell divisions until Q1 comes on. And just for full disclosure, we don't have a complete answer to this, but I will tell you what we do know. And I think these are important points that will lead us forward. So the first thing that we wanted to know about the maintenance of the Lausy-6 competent or primed state was whether it required additional asymmetrically expressed genes. So you can hopefully envision a model in which TVX3738 prime Lausy-6, but in addition to that, they activate the expression of other genes, potentially other transcription factors that might either maintain that asymmetrically primed state or even act at the end, together with Q1, to activate Lausy-6 only in the left ASC neuron, but not in the right ASC neuron. Alternatively, TVX3738 might really only just prime Lausy-6, and then none of their targets might be necessary, and this may just be maintained by other mechanisms that may or may not include other factors, as we will discuss later. So the experiment that we did was to try to see if an asymmetrically primed Lausy-6 locus can be maintained in the absence of TVX3738. So for that, we started out with a TVX3738 mutant, complete mutant. This animal, as I showed you, is morphologically abnormal, but also it generates ASC neurons that do not express Lausy-6 at all. And one just small note, you will notice 3 ASC neurons. That's because due to some lineage conversions during this developing embryo, sometimes they produce 2, sometimes they produce 3 ASC neurons. But that's just a small detail. So in this animal that does not have TVX3738, we asked if we now asymmetrically prime Lausy-6, could that be maintained in the absence of other asymmetries in the lineage? And for that, we used the heterologous system that I just introduced, in which we use the Lausy-6 GFP locus that has UAS sites, and we provide the activator GAL4-VP64 using a TVX37 promoter. So now we have an asymmetrically primed locus, and we ask what happens, is Lausy-6 expressed in these cells or not? And what we found is that in practically all embryos that we looked at, Lausy-6 is indeed expressed in one of the 2 or 3 ASC neurons that are made in these embryos, even though TVX3738 are not there. So what we think this experiment tells us is that this asymmetrically primed state of Lausy-6 can be maintained in an otherwise symmetric transacting factor environment. So the transcription factors that are present in both these lineages now in the TVX mutant are the same. and yet the asymmetry can be maintained in that symmetric environment. So we think this is very interesting, and this tells us that if other transcription factors are required to propagate that active state of LAU-C6, they must be symmetrically or even possibly ubiquitously expressed, but they will just propagate the signal where the initial trigger happened. Now, I have been talking about accessible DNA and essentially propagation of competent states without really going in a lot of detail about chromatin, and that's because we don't know a lot about what happens on the chromatin. By chromatin, I mean histone modifications on the LAU-C6 locus. But I will tell you what we know. Julian did a screen for chromatin modifiers, for histone, primarily histone modifiers that are important for the expression of LAU-C6. And these actually complemented other screens that were done in the Hobart lab, primarily led by Richard Poole. So this was an RNAi screen, and I will not go into detail about the different factors that Julian found were important. I just want to focus on these genes that are all part of the compass set1 compass complex. I'm showing you here the composition of this compass in Drosophila, but it's essentially the same in C. elegans. We find really practically all components of this complex are important for LAU-C6 expression. This complex methylates histone 3 on lysine 4, and this is a mark that is associated with activation. So this is very interesting. It seems that you need to establish this active transcriptional state, and you need, in order to either establish that or maintain that state, you need H3K4 methylation. However, of course, this, as I mentioned, this is an RNAi screen. This is a loss of function of this complex across the whole animal. So we could be affecting LAU-C6 expression through any sort of indirect measures. So I'm very pleased to show you this more recent experiment that Julian has performed, in which we asked, or we are able to finally assess whether this mark is required on the LAU-C6 locus itself. And for that, we again use this tethering system, and we now have a LAU-C6 locus in which the GAL4 binding sites are introduced in an intron of GFP. That locus has wild-type TBX37 binding sites, wild-type Q1 binding sites, and so in the absence of any tethering, this locus is perfectly expressed in the left ASC neuron of every animal we look at. However, if Julian now tethers lysine 4, histone 3 lysine 4 demethylase, he observes that in a fraction of animals, he completely loses LAU-C6 expression, and in another fraction of animals, a very large fraction of animals, the expression is very, very low. So there's two different demethylases in C. elegans. There's SPR5, which is thought to be primarily a dimethyl demethylase, while RBR4 is thought to do both di and trimethyl demethylation. So RBR4 has clearly a stronger effect in this assay. So this is very exciting, because we think what this means is that H3K4 is necessarily on the LAU-C6 locus, and now we can play with timing and try to address when is K4 required. And so these are all experiments that Julian is performing, hopefully not as we speak, because I think he's watching the talk, but maybe soon after. So let me summarize what I've told you so far. Essentially, what we think enables the interaction of these two transcription factors over time is that first of all, these T-box transcription factors bind directly to the locus, promote transcription, make the locus accessible, but then they're really not required anymore on the locus. Somehow that active transcriptional state is maintained for 4 cell divisions. We don't fully understand how, but we know that that maintenance does not require other asymmetric factors, but possibly other transcription factors that are broadly expressed, and very likely also chromatin factors that are helping to maintain that active transcriptional state, such that when Q1, this second transcription factor appears, it is able to boost transcription to the needed functional levels from this locus. The other thing I didn't go into, but the timing experiments for requirement of TBX I showed you suggest is that the the LAUSI6 locus beyond a certain time window adopts a repressive state that prevents activation by key 1, for example, in the right ASE neuron. So we think that this priming event either prevents the onset of that repressive state or turns a repressive state into an active state. This is something that is still also an active area of investigation. But I would like to now move a little bit away from the mechanism to the implications for this. And basically what I've been talking about is this pair of neurons, the left and the right ASE neurons, which are made asymmetric by these asymmetrically expressed early transient transcription factors. However, as I also mentioned at the very beginning, these parts of the lineage of this sublineage of ABA and this sublineage of ABP give rise to a number of neurons that occupy left-right positions in the head of the animal and also have this asymmetric lineage origin. So this might also be subject to priming by TBX, not on LAUSI6, or presumably not necessarily on LAUSI6, because LAUSI6 is only activated later on by key 1, but potentially on other genes. So we set out to explore that, and the first gene that Julian focused on is the gene that is downstream of LAUSI6, because we assumed that it shared that TBX binding site with LAUSI6. That TBX binding site can also presumably affect transcription of that downstream gene. And in fact, looking at expression of that gene, we indeed saw that it's asymmetrically expressed in the ASE neurons, but it is also present in a number of nuclei on the left side of the head, but not on the right side of the head. So this was very exciting because it could be potentially other neurons with this asymmetric lineage origin. And indeed, we went on to identify what some of these neurons are, and I'm showing you here now specific markers, for example, for the AVE, left and right neurons, in which you can see this gene is expressed exclusively on the left, but not on the right side, or the AUA neurons, again, left and right, in which you can clearly see expression on the left, but not on the right side. So what we think this tells us is that this priming, this early asymmetric priming, might potentially be affecting other neuron pairs. There's a number of candidate genes that we have now to follow up on and try to address whether they are indeed asymmetrically expressed and whether that has any functional consequences for the function of these neurons. But with that, I want to generalize this maybe a little bit more and try to discuss in what context do we think that this is important. And maybe before I go into that, there's one point that I would like to make, which is that there are many instances that have been described in multiple different contexts in which a locus needs to be primed for activation before its actual transcription. There are many poised loci, poised enhancers, and people have called this by a number of names. But in all of these cases, I would argue the priming has been thought of or has been interpreted in the context of producing robust or timely transcription at a later time point. For example, priming is thought to be used to overcome the rate limiting step of remodeling chromatin and making a locus ready for transcription whenever that transcriptional activator comes, such that the locus can produce a rapid response. So these are all mechanistically extremely interesting possibilities. But what I would like to propose is that we can reinterpret some of these priming events in the context of using them as to produce temporal combinations of transcription factors. Because priming in this case, it's not that the LAUSI6 locus needs to be rapidly activated at a particular time point, but I think in this case, the priming event determines whether this locus gets activated or not at all in response to a symmetric terminal differentiation problem. And so I think such a mechanism could be very interesting to explore in this context in which two cells are, or more cells, are basically are made to execute the same terminal differentiation program, but they derive from different trajectories or different histories. And that is what we call convergent cell types. And there's plenty of examples of these in the C. elegans lineage that can be observed. But this is also a very widespread phenomenon in the development of every animal. And there are some examples that I list here. One of the most prominent ones is perhaps cell types that derive from the neural crest cell, but also from mesodermal lineages, and in the end give rise to very similar cell types. So what we're proposing here is that these cells that achieve very similar terminal identities through different paths very likely carry differences from their different trajectories that will make them slightly different, and this can be a mechanism to generate broader diversity. Okay, so with that, I'm wrapping up, and I just want to sum up what I discussed with you today. I first showed you our efforts to understand the mechanism by which the Lausisix locus is primed by TBX3738 for the later action of Ki1, which is separated by 4 cell divisions. I also try to give you an idea at the end about what the implications for this mechanism are directly in the worm, but also more generally in thinking about how combinatorial expression of transcription factors is used during development to generate cell type diversity. And we are going to continue to, but we are also excited to see how people think about this idea of temporal combinations of transcription factors in analogy to the spatial integration of transcription factors that is generally used to generate cell type diversity during development. We're excited to see where this leads us in the future. So with that, I will finish by thanking and acknowledging the people who did the work. As I mentioned, this was a team effort led by a fantastic PhD student, Julien Charret, and a previous postdoc, Thomas Daniel. There were several people involved in the study. I highlighted the ones who had the major contributions, but there, so as I said, Alex and Ariane were involved in all the ATAC-seq experiments. Jinqui did all bioinformatics analysis, and Mila generated lots of strains for this project. But this project also had wonderful contributions from Josef Resner and from someone some of you might remember over there. Paula Gutierrez Perez did some beautiful microscopy. She was a master's student at your institute, and she's she has a fantastic story on microRNAs I didn't get to talk about today. And yeah, thank you for your attention, and happy to take questions.
Okay, you can hear me now, I guess?
Perfect, yes.
Thank you very much. It was really, really nice talk. You have a couple of questions.
Mm-hmm.
So Fernando Martin, he says that, well, thank you for the talk. It was a beautiful work. And then he asked, okay, so whether you believe that the system in the right neuro is defined by another specific priming mechanism, or whether is it a default? I mean,
yes.
Redefining the question, does ASER cell also require to be primed by a different equivalent to TBX 3738? Yeah,
that's a very good question. The answer is that I don't think so, and this is because in the absence of Lausisix, so in the absence of TBX 3738, and therefore in the absence of Lausisix expression in ASCL, this cell adopts an ASCR fate. So we know that Ki1 expressed in those cells is able to drive the ASCR fate. It will only execute a different fate if Lausisix is also there. And that's because Lausisix acting as a repressor will repress other downstream factors. But we also know from experiments from another colleague of ours, Barish Tursun, that if you ectopically express Ki1 in other cells, even in germ cells, you actually activate expression of genes that are normally expressed in ASCR. So I think we have enough lines of evidence to suggest that the ASCR is a default fate that is triggered by expression of Ki1. So that's what I would say to that.
So sorry, I think you cannot hear me very well, right? It's very
no. For me, it's okay.
Okay. Another question. Jose Antonio Esteban also congratulates you on your talk. And he asked, the Lausy-6 locus eventually closes. How does this happen? And how the timing of closing is established? He means the availability afforded by TVX is closed, perhaps shifted to Q1. How would this happen?
If the second part of the question I didn't quite get.
That basically is a refinement of the question. If the availability afforded by TVX is closed, perhaps shifted to Q1, how would this happen? I guess this.
Okay, so I can go for the first part of the question. The second part I'm not sure I understood, but I can maybe it gets clear with this. So there is definitely a repressive state that is that sets on the Lausy-6 locus. Now we assume this has to do with repressive histone modifications. In C. elegans, there's no DNA methylation, for those of you who might be thinking about this. So we know it cannot be cytosine methylation, but there are all the repressive chromatin marks that people working with other systems are familiar with. So there's methylation of lysine, histone 3 lysine 27 by the polycomb complex. There's also methylation of histone 3 lysine 9 by other methylases. So we have tried to use genetics to see if any of these complexes are what keeps or what represses the Lausy-6 locus. Essentially, we looked at mutants for polycomb or RNAi. We looked at mutants for different H3K9 methyltransferases, and we haven't found any single complex that does the trick. However, we have some pretty exciting preliminary data from Julian that suggests that the HP1-like proteins, which are proteins that bind methylated H3K9, are actually determining this window of opportunity during which TVX37 need to act. So effectively, if Julian gets rid of 2 HP1-like proteins in C. elegans, that window extends. And now TVX are able to act at later time points. But that is as far as we have gone on this. But now we have the tools with the tethering and so loss of function, gain of function to really try to get deeper into these questions.
Okay. Another question by Carlos Estella. Great talk, awesome work. The question is regarding the role of Lausy-6 transcription, sense and antisense, in the chromatin accessibility and binding of Q1.
Yes, so we haven't done, so essentially the question would be if we have done a TAC-seq in, so we have done a TAC-seq in animals in which we deleted the TVX binding site. And there we don't see either transcription of Lausy-6 or binding of Q1 indirectly by looking at accessibility of the Q1 binding site. But it's a difficult question to address experimentally. I think we have done, so playing with the transcriptional activator and the transcriptional repressors, we suggest that transcription is required, but we don't know exactly how it's required. So I don't know what exactly Carlos is referring to, but there's a different possibilities for how transcription might make the Q1 binding site accessible. And that might be simply because polymerase going through the locus just displaces nucleosomes and makes the other binding site accessible. It might be due to bringing in different chromatin modifiers, such as the set1 compass complex. But I'm sorry, I don't have a much more intelligent answer to give. This is essentially the state of what we know at the moment.
Okay. I also have a question about the relevance of your very exciting findings in the asymmetry of other neurons. I guess you don't know what the C34, C4 gene is, but because this is very interesting, right? Like we struggled to find different asymmetries in the nervous system, and suddenly, it's more common than expected. Are you doing some functional assays on these cells to?
Yeah, so there's a few things about this gene that make us think that it may or may not have a functional consequence. But regardless of this particular gene, there's a set of about 100 genes that we see are bound by TBX3738 in the precursors of the left neurons, and obviously not on the right side, and that remain differentially accessible by a TAC-seq between the AV and AVP derived. So there's a bunch of other candidate genes that I think are more relevant to study. However, we are still doing some experiments on this gene. The problem with this gene is that it's extremely short. It's only 44 amino acids. It has a transmembrane domain. It may be a real gene. It may be a remnant of something. It is clearly expressed. It is transcribed and translated, that we know. But we will delete it, and but we have really no clue where to start looking for functions of this gene, right? So we will do behavioral assays and assays to see if the sensory neurons are functional in the context of what we know they do. But I think it's a bit of a needle in a haystack problem at the moment. We will try, but I don't know if this particular gene will be the key to answer that. But we have other exciting candidates as well. And I'm not sure that asymmetry is so common, but in this context, so not all neurons from the C. elegans nervous system are generated through these asymmetric lineage origins. So it might be a property of neurons that arise through this type of development.
Because I was also thinking, how prevalent do you think this temporal intersection is going to be? Like, we have cases in whereby the combination of transcription factors, we cannot really differentiate terminal sulfates.
Yeah.
Those cases are not as, I would say, technically easy to manipulate versus ASC, but it could be something similar to this, right? Like one cell type is primed.
Yes.
But I don't know if you think how to tackle that.
So in C. elegans, we are actually going forward. In other systems, I think it's clearly experimentally more challenging, at least for my skills. But in C. elegans, we are indeed addressing this. And the way we started looking at this was to look for other similar instances of cells that seem to acquire the same terminal identity, but do so through different trajectories. And actually, we're zooming in on muscle. So we are getting very, very excited about the body wall muscles because they have extremely interesting, very different lineage origins, and they converge on the same cell identity. And what we have now is single cell RNA sequencing for a lot of these lineages. And we know that, that these muscle cells are actually not identical at the end. So there are differences, and now we can try to trace back, do they depend on those early differences in their lineages or not? So that is how we are continuing to study this.
All right, well, I think that's a good outcome. Thank you very much for coming and give us such a beautiful talk. I don't really know how to end this. So.
I will do stop share first.
Yeah, okay.
And then
okay.
Should I leave?
Okay, let's, yeah.
I will leave and see you later.
Yeah, okay.